Here is Tom Blumenthal, and Tom Blumenthal is another triple threat. He is not only an incredible molecular biologist who's tackled some of the most important questions of gene expression, but he also has been an administrator, probably a really ugly word, because he's been the chair of 3 different departments at 2 different universities. And finally, he really works on his passion of helping people with Down syndrome, and I think it's this last one that he's going to be concentrating on today. Thank you, Dr. Blumenthal.
Thanks. Nice introduction. Actually, I try to think of myself as a credible molecular biologist. So if you want to get a grant from the NIH, you have to propose a particular idea and then some experiments that are definitely going to answer the question you've asked. The worst thing that can possibly be said about your grant during the study section where we decide who gets the money is that it's a fishing expedition. That's the kiss of death. And I want to say that omics is all a fishing expedition. So I've never heard before about Larry's proposal. Didn't hear about it until today. But I want to show you that if you actually engage in some fishing expeditions, like Larry's massive proposal is a fishing expedition, you can catch fish. But if you don't do it, you won't catch fish. So that's my title, but not my title slide. Here's my title slide. And so I'm going to tell you about 2 fishing expeditions today, but first I'm going to talk a bit about the river in which we're fishing. So that's Down syndrome. So you just heard that 4 years ago I was the most basic of basic molecular biologists. I was studying RNA in worms. I was chair of this department here, molecular, cellular, and developmental biology. But now I head the Cernic Institute for Down syndrome. And I just want to tell you, I never once thought about Down syndrome until I took this job. So I'm going to tell you why. So Down syndrome is, in fact, a fascinating but understudied and misunderstood medical puzzle. So just a little bit of background. 1 in 750 live births in the US is born with Down syndrome. That's more than 300,000 people living with Down syndrome in here and millions worldwide. The average life expectancy of these people is now more than 50 years, nearly 60 years. Did not used to be true several years ago. So what is Down syndrome? It's the most common genetic cause of cognitive disability. IQs range typically from 30 to 70. And then there are developmental abnormalities with which you're probably all familiar. Many of you probably don't know that people with Down syndrome get Alzheimer's disease at very high frequency. I'm going to talk a little bit about that in a minute. They get leukemia at relatively low frequency, but much higher than the general population. And that's true of all of these childhood leukemias. They get autoimmune diseases at a high rate, but they get solid tumors at a very low rate, much lower than the general population. They also get cardiovascular disease at a low rate. And then there's more that I don't have time to talk about. What causes all of this? A single extra copy of chromosome 21, the smallest human chromosome. So you're supposed to have 2 of each, but people with Down syndrome have 3 of this little one. So it's only got a few hundred genes. So how does an extra copy of 300 genes out of the 20,000 total do that? So there's no mutant gene, and this is an unusual genetic condition, right? There's no mutant gene. It's unlike other genetic diseases. So is there a regulatory imbalance that causes this? And if so, basically that has to be the case, which regulatory alterations matter? So how might an extra copy of chromosome 21 cause Down syndrome? So there's a variety of ideas out there. 1.5-fold overexpression of one gene could do it, or a few genes. Or maybe one gene does one thing and another gene does another thing. Another possible idea is that chronic induction of 150 proteins could cause a kind of stress response, oxidative stress or proteotoxic stress, just by having that extra chromosome. And this could be responsible for the symptoms. And certainly some of the symptoms involve repression of cell cycle entries. So the cells have to divide, and these cells don't seem to divide as easily as cells of typical people. And if those are stem cells, failure to divide instead of differentiating could cause a lot of the symptoms that are seen. I also want to mention that there's a very high variability of symptoms in Down syndrome. So some people get various things much worse than other people, yet they all have the same extra chromosome 21. This could be caused by alleles on chromosome 21, so extra copy of some gene on 21. It could be the genetic background, and it could be environment, perhaps environment in the womb, since this is a developmental issue, and it could be bad luck or good luck. So just to emphasize, people with trisomy 21, Down syndrome, have a different disease spectrum. They are unlikely to get heart disease, cancer, stroke, etc., and much more likely to get Alzheimer's, leukemia, autism, epilepsy, congenital heart defects, which can be and are cured by surgery. So I just want to point out that many aspects of Down syndrome open interesting and tractable biological problems. And so obviously the research we're doing is going to be important for people with Down syndrome if we make advances. And it should also provide a deeper understanding of gene regulation, which is one of the things that attracted me to the field in the first place. Let me say a few words about the Alzheimer's connection, because it's one we actually understand at some level. So people with Down syndrome provide a unique population where everyone has Alzheimer's pathology, plaques and tangles, and most will get Alzheimer's at very relatively young ages. So this is an opportunity to study Alzheimer's disease before it shows symptoms, because in you and me, that's not possible, because we don't know which one of us is going to get it. But these people are all going to get it. So, and we know what's the cause. And this is the only case where we know the cause of a particular symptom of Down syndrome, because on chromosome 21 is the gene for amyloid precursor protein, APP, and it's overexpressed 1.5-fold, we know, which is clearly the reason that people with Down syndrome get Alzheimer's disease. It's the only case we know of. So obviously, solving Alzheimer's disease would be an enormous contribution that the Down syndrome community could make to the population at large. And of course, doing so would be an enormous benefit for those with Down syndrome, right? A word about the leukemia connection. It's a very interesting one. Why do people with Down syndrome get childhood leukemia at high frequency? We don't know the answer to that at all, but there's this very interesting observation. So there's this very rare childhood leukemia called AMKL, which has a much higher frequency in Down syndrome. And people with that all have mutations in this transcription factor called GATA1. What they discovered is that people without Down syndrome who get AMKL also have the GATA1 mutation. But they have acquired trisomy 21 in the cells that become leukemic. So that shows a direct connection between people with Down syndrome and the whole population. If we could come to understand it in one, we'd come to understand it in the other. Why do people with Down syndrome get so few solid tumors? And there's a lot of good ideas, all of which are possible, and we need to figure out which one's right. So I mentioned the stimulation of the stress response. So one possibility is when the stress response, oxidative stress, is stimulated, that may stimulate suppression of tumors. Second possibility is that, in fact, they have the same number of tumors as everybody else, but you don't know it because the tumor cells fail to divide. So maybe it's not less cancer. Maybe it's just the cancer doesn't hurt them. Is there a key tumor suppressor on chromosome 21? There might be. One possibility is this gene endostatin, which is on chromosome 21, which is known to suppress angiogenesis. Angiogenesis is the attraction of blood vessels to the tumor. If the tumor can't get blood, it can't grow. And this is another possibility, which is 2/3 of the people with the extra chromosome 21 never get born. It's possible that the ones that do get born are more robust in some way than those that don't, and so they're less likely to get cancer. It's an idea. So what signaling disruption is responsible for the cognitive disabilities? We don't know. When we do know, soon I hope, can we intervene? This is supposed to show a mouse looking for a way out. So clearly, the study of Down syndrome is potentially very valuable. Hopefully, I've even convinced you at this point that people with Down syndrome represent, in fact, a gift to biomedical research. And we hope to make some progress in doing that, and I'm going to tell you about 2 fishing expeditions now. One is, they're both omics. One is proteomics of blood from people with Down syndrome, and the second is transcriptomics of cell lines from people with Down syndrome. And this is a report on a project done by a collaborator of mine, Joaquin Espinoza, who also used to be in this department, but is now at the Cernic Institute for Down syndrome, and is, in fact, is our associate director. So the proteomics project, first, we're doing this with SomaLogic, which I assume has something to do with the fact that I'm up here talking with you today. Maybe not. I don't know. No. Larry says no. So we're measuring protein levels in blood from people with Down syndrome. We're hoping we can identify clues as to how various symptoms are caused, what changes correlate with which symptoms, which ones predict severity. I'm not going to tell you about any of that today because we're in process, but I'll tell you about this last one, is to identify biomarkers and potential drug targets. So I'm not going to go through the SomaLogic's technology, but just to say that we're doing this in collaboration with them. They have made these DNA aptamers that recognize particular proteins so that they use these to pull out the proteins and then tell us how much of that protein is present in this blood sample. That's the only question they're answering. How much is there? In fact, if you look at people with Down syndrome and compare them to controls, are there different amounts of some proteins? And if so, which ones? So I'm going to talk about 3 studies, but mainly only about one. And these bars show the number of subjects. So we compared about 120 people with, who have Down syndrome with about 50 who don't. And we're looking in this study at 3,600 different proteins. And then these other two studies are smaller studies just to find out if we find something interesting in this study, is it reproducible in these two studies? Because they're different people. Do we get the same answer? So here's a lot of dots for you non-scientists, and I'm going to explain for you what these dots mean. The scientists may well be aware of already. So let's look at this one. Every dot is the level of this protein, fibroblast growth factor receptor 1, the amount of that protein found by SomaLogic in the blood of one person. Every dot is. And this line shows the people with trisomy 21, and this shows the people who are controls in our experiments. And what you can see quite clearly, I hope, is that this red box is higher than this blue box. And in general, most of the dots associated with the red box are higher than the ones associated with the blue box, saying the level of this receptor is increased in the blood. And if you look at this is another protein, same thing, measured at the same time by SomaLogic. And what you can see is this epidermal growth factor receptor is lower in people with Down syndrome in general. Okay, that's all we're measuring. So this is called a volcano plot from the same data, and you need to understand the volcano plots because I'm going to show you a bunch of them. On this axis shows you how significant the results are. So here's FGF receptor. It is, in fact, the most significantly different one we found, significance being a statistical measure of whether the difference is real. And then this blue line shows no change, and on the right of this line, at least to me, on the right, no, to you too, increase, and here, decrease. So you can see, first of all, from this, that a lot of proteins are statistically significantly different in Down syndrome. Some of them go up, and some of them go down. And these lines is an arbitrary place called a false discovery rate of 10%. So 90% of these are definitely different. So here's the 3 experiments for fibroblast FGFR1. The data I showed you already and the two validation studies. And you can see that we got the same answer every time we did this. That's not always true, but it's true for a bunch of them. I'll just whip through a few slides here. This shows a different way of plotting the same data. This is the FGFR1 data. But in this case, we've made it so that every dot in every study is a different person. We did a sort of longitudinal study on some of these people, but we took out all of the duplicates. So none of them are duplicated, and you can see when none of them are duplicated, you get the same result every time. Here's a different protein, cystatin C. It's up in Down syndrome. Here's a different protein, beta-2 microglobulin. It's up in Down syndrome. And these are all 3 studies. Another one, MMP1, it's a little harder to see, but in fact, this one's way up in Down syndrome. TFF3, this gene is on chromosome 21. It's up in Down syndrome. You don't care, right? But it's a bunch of them. This one's called coagulation factor X. It's one of the coagulation factors, as you might have guessed, and it's down in Down syndrome. Antithrombin 3, it's down, and it's down every time we look at it. So I think we can be quite confident that the results we're getting from this analysis are real. So here's the data I showed you already. Lots of changes, and if we just take the same samples and we divide them up into males and females and ask, does that give us the same thing? The answer is no. So there's almost nothing significantly different between males and females, whereas there's loads different between people with Down syndrome of either gender and controls. Most of the proteins that are changed So this shows the same data I've been showing you with some numbers. So when we look at all the proteins, 121 of them are up in Down syndrome and 178 are down. If you now look at the red dots on the right, you can see that of the 50 chromosome 21 genes, some of them are different, but most of the ones that are different aren't on chromosome 21, and the ones that are on chromosome 21 are all up. None of them are down. That's what you'd expect, right? There's an extra copy. Everybody's on top of that. And here's a different way of showing the same data. This is it's called a Manhattan plot. And you can see that on this chromosome, some are up, some are down. But on this chromosome, chromosome 21, they're all up. And in general, genes on chromosome 21 are up 1.5-fold, just like you'd expect. And I'm saying the protein levels in the blood are, because that's not an obvious thing. It wouldn't necessarily be true, but it turns out to be. So now the key question is, do the protein changes potentially explain some aspects of Down syndrome? And I'll just go back to FGFR1 again. Here's FGFR1 by age. And you can see that virtually at every age, people with Down syndrome in the triangles have higher levels than people without Down syndrome. And this is the same data you've been seeing. So there happen to be some mutations in the human population where FGFR1 is increased. These are mutations in the FGFR1 gene that result in higher levels of FGFR1. I'll show you 3 of them. One's called 8q11 myeloproliferative syndrome, which has enhanced signaling in the bone marrow. That leads to myeloid expansion, which develops into one of the childhood leukemias, which is up and Down syndrome. These 2, osteoglophonic dysplasia and Pfeiffer syndrome, both alter bone growth in such a way that results in distinctive head and facial features, dwarfism, abnormalities in the skeleton, which resemble Down syndrome. So one possibility is that the increased level of FGFR1 is relevant to symptoms of Down syndrome. Nothing we were looking for. This was a fishing expedition, and we think FGFR1 is a fish. So a few other things, I'll just run through them for the scientists in the audience who may find it interesting. 3 EGF receptors are down. So EGF receptors are involved in growth, cell growth. And the levels are lower. And these 2, at least, are activated by neuroregulins, so they're important in development of the brain. Neurotrophin receptors, also involved in brain receptors, TRK-B and TRK-C are both down in Down syndrome. And these are important receptors for a protein called BDNF and nerve growth factor. So this may be a very important finding. We have evidence that kidney function may be impaired. I've never heard of that in Down syndrome, but proteins that go up in blood often go up in blood because kidney function is failing. And these are 2 that are often used as measures of glomerular function. The complement cascade for blood clotting is down. Those are all different members of the coagulation cascade. I'm sorry, of the complement cascade, important in the immune system. The coagulation cascade is also down. Neuropilin, a very important protein, it's a receptor for VEGF. VEGF is a protein needed for angiogenesis, which I talked about a little while ago, and semaphorin, which is important in nerve cell identity. So this is very important in axon guidance and blood growth. This thing's up. So this is plotted a different way so you can see it. Most children with Down syndrome have extremely low circulating IgE. Now, IgE is involved in allergy. This wasn't known until we did this experiment. We get the same result every time. So what is shown here is the IgE levels measured by SomaLogic on this axis and the percent of subjects in each of these bins. And it goes from 300 to more than 50,000 out here. And what you can see is in the typical population shown in blue, that it's all over the map. IgE levels are different in everybody. Whereas people with Down syndrome, almost all of them are in these lowest bins. We don't know what this means. We would predict maybe that they would have less allergy. But I've never seen anybody do a kind of experiment that asks, do people with Down syndrome get less of something that's not that important? Nobody, so as far as I can tell, nobody knows whether that's true or not. So we still don't know, how does the extra chromosome cause these changes? Except for the one I mentioned, APP, we know how it causes Alzheimer's. And what are the consequences of these changes for people with Down syndrome? That's going to require some more work. So my favorite hypothesis, and I'm going to give you two versions of this hypothesis, one before Joaquin Espinosa's transcriptomics experiment and one after. Here's the before. The extra chromosome causes global gene expression changes, resulting in direct and indirect production of extra proteins. This induces the stress response, resulting in suppression of cell division and senescence or premature differentiation of dividing cells. So stem cells senesce or die, resulting in altered levels of different cell types in the blood, and the protein level changes that we see could be a consequence of this alteration in the relative abundance of different cell types. It's a hypothesis. And so I'm going to move to our second fishing expedition, but first I wanted to mention the people who've contributed to the work I've showed you so far are here. These are people at the Linda Cernick Institute for Down syndrome, and the people at SomaLogic who have contributed and helped us, and the people at the C Center who have been instrumental in us being able to get the samples. And of course, we're grateful to the children who gave the blood samples and their caregivers, and the funding from Anna and John J.C. Foundation, the Global Down Syndrome Foundation, and both campuses of the University of Colorado. So we couldn't have done this without that because it was a fishing expedition, and NIH doesn't fund them. So now let me tell you about the Espinosa project. So first, the generation of this project. Where did it come from? So what we do at the Cernick Institute is we give out grants to people on the Boulder campus and the Anschutz Medical Campus and at National Jewish Center in Denver to initiate projects on Down syndrome. So scientists are always looking for money, and we've given out, I think, more than 30 different people have gotten money to start projects, and some of them are in this room. One of them was Joaquin Espinosa when he was a faculty member in this department, MCDB. And he started this project associated with that grants program. So that grants program has had mixed results. We've had some fabulous projects that have made real advances, and some that have simply proven that their hypothesis was wrong. But I think this way of doing science is great. So what's Joaquin done? So his object was to identify the key signaling pathways consistently activated by trisomy 21. So this isn't proteomics like I've been showing you. This is transcriptomics. He's going to look at the levels of messenger RNAs in cells, of trisomic cells, and compare them to disomic cells and ask, what's different? And so the idea here is to measure the important thing. So this shows trisomy 21, 3 chromosomes, stimulating something, and that something is going to result in a whole lot of downstream effects, and he's going to figure out which they are. So it may actually stimulate many things, but he's really looking for the biggest blue circle, that is, the biggest signaling event that's altered. So his approach, as I mentioned, is to do transcriptomics to elucidate the signaling cascades. And he's doing this measuring 16,000 different messenger RNAs by RNA-seq, for you aficionados. And he's doing this on human skin fibroblasts first and lymphoblastoid cell lines second. So he's doing the same thing with two different, completely different kind of cells. Fibroblasts are skin cells. So I'm going to show you that first. So he's comparing 6 cell lines, disomic, with 6 cell lines that are trisomic. And these people are unrelated to each other, any of the 12. They're all different, and he's looking at different ages, genders, biopsy sites, all kinds of differences, and he's looking for what's consistently different. And this just shows that these people here really did have trisomy 21, and these people really didn't. So again, I'm showing you a volcano plot, and what you can see here is that there's a lot of genes that were consistently different comparing trisomy 21 versus typical. And the green ones are on chromosome 21. So again, we see that chromosome 21 genes are up about 1.5-fold, just what you'd expect for an extra copy of the gene. So then the question is, that's only about 12% of them, of all the genes that are changed. How about the other 88%? So those have to be changed by some signaling difference between chromosome 21, trisomy and disomic, right? So some of them go up, 471, and some of them go down. Of the 471 that went up, 81 were on chromosome 21. Of the 191 that went down, none were. And this shows you the other kind of plot I've been showing you, the box plot, showing that genes on chromosome 21, they go up, but the distribution of people is similar. Now, we're only comparing 6 and 6 here, so the statistics isn't nearly as good. But he typically sees this, which is that the disomic cell lines don't have a very large spread, but the trisomic cell lines do. So there's much greater variability. We don't know what is responsible for that variability. So now then the question is, what's changed? What's done that other 88%? So he looks by various pathways. One of the ones he's looked at is ingenuity pathway analysis, which asks the question, for all these genes, what upstream regulator might have changed their levels? What's different? And that's essentially the question, what's the blue circle? And the answer is interferon. So some of you know what interferon is, some of you don't. It's clearly the biggest blue circle. We had no idea this would be the case. This was completely a fishing expedition, and interferon is the fish. So I'll tell you about interferon in a minute, but there's 3 types of interferon, types 1, 2, and 3, and interestingly enough, nobody has paid any attention to this, of the receptors for interferon, there's 6, 4 of them happen to be on chromosome 21. In fact, there's a single cluster of genes containing interferon receptors. So interferon receptors act through this group of kinases on this group of transcription factors, which then regulate all these different genes. And so he's found that, so this shows the activation or suppression of the top group of genes. So this is the top group. This is the one he found as the most changed, all of them non-chromosome 1s and chromosome 21 ones. And what you can see is these interferon receptors are in fact overexpressed. All the ones on chromosome 21 are overexpressed. This one's somewhat underexpressed, but it's not on 21. And this one is really not very different. So what is interferon signaling? So interferons are signaling proteins made in response to the presence of tumor cells, but mostly pathogens, viruses. They are cytokines, proteins used for communication between cells to trigger the protective defenses of the immune system. Related functions include activation of immune cells, such as natural killer cells and macrophages, and upregulation of antigen presentation by increasing expression of major histocompatibility complex antigens. And chronic expression of interferons is harmful to the organism, so they're turned off as soon as they're no longer needed. And that's a key, because the people with Down syndrome are suffering from chronic induction of the interferon response. So this just shows how many of these are related to interferon. So all of these ones with the red arrows are involved in the reception of the interferon molecule, or they are the interferon molecule. These three are parts of the signaling protein. The signaling transcription factors. And then there's lots of the ones in the bottom box, which are interferon responsive genes, which are changed. So there's no question about this signature. It just jumps out. If you do the same thing in the lymphoblastoid cells, you get the same answer. So he did that, but not as robust a set of experiments. He's compared only 3 to 3. Some were up, some were down. Chromosome 21 ones are up. And if you look at what he got the same thing again. The interferon response just came out again. So these are actually white blood cells. And then this shows more of his evidence. Some of it is based on his RNA-seq, and this shows that a Western blot shows the levels of the protein products of these genes, shows the same increase that you would expect based on their messenger RNAs going up. So his conclusions are that trisomy 21 causes a consistent gene expression signature that withstands inter-individual variation. The non-chromosome 21 fraction of the signature is highly enriched for interferon-inducible genes, and this could be explained simply by overexpression of 4 of the 6 interferon receptor subunits encoded on 21, although we don't know that it is. I'm just saying it could be. So this would result in induction of the JAK-STAT signaling, and this would be sufficient to explain the cognitive deficits, because during development, progenitors of neurons and glia, which are support cells, there's always a choice made between any of those cells. It's going to be a neuron or it's going to be a glial cell in the brain. And interferon is known to affect that choice. And in fact, in Down syndrome, we know that there are fewer neurons and more glia. So that could be done by interferon. We don't know that it is. It could explain the leukemias. We don't know that it does, but it certainly could, as I explained earlier. The great variety of autoimmune diseases could be a result of the increase in interferon, and so could suppression of the solid tumors, because interferon is known to suppress solid tumors. So an implication of this is that Down syndrome could be classified as an interferonopathy, along with other genetic conditions caused by gain-of-function mutations. So it's possible that the ill effects of Down syndrome could be ameliorated, even perhaps eliminated, with available JAK-STAT inhibitors and other inhibitors of the interferon pathway. And there are many drugs made by these companies that work at this level, or this level, and these could be tested. And now will be. And here's an example. Alopecia. So alopecia is an autoimmune disease, and it can be treated with ruxolitinib, which is an FDA-approved JAK inhibitor already on the market, and you can see what it does for these people. Cures them. So there is already an interferonopathy, or a group of interferonopathies. This little girl, Jabina, suffers from one of them. It's a group of genetic disorders characterized by upregulation of the interferon response. And here's the symptoms, and a lot of those symptoms are quite similar to symptoms of Down syndrome. Cancer patients are often treated with interferons. And I just want to let you look for a second at the comparison between known interferon side effects and Down syndrome. So here's my modified hypothesis. The extra chromosome causes global gene expression changes due to interferon signaling and induction, which causes profound changes in the immune system, including systemic inflammation, and that results in all the rest of what I had there on the first slide. That's a perfectly reasonable idea. We don't know if it's right yet, but we're gonna. Now, I know you're sitting there wondering, if this is so clear, how in the world could this have been missed all these years? And the answer is, it wasn't missed. The key role of interferon signaling in Down syndrome was first reported in 1974 in a paper from Frank Ruddle's lab at Yale. And for reasons that are not clear to us, it's been ignored by the field. And it continues to be ignored by the field. It's a very strange field. So this is that paper, and many of these people went on to stellar careers, like Charles Epstein was a very well-known human geneticist. Jay Tishfield, Chris Tan. Epstein was actually a target of the Unabomber. So anyway, he showed this a long time ago, and I'm running out of time, or I've run out of time probably a long time ago. But then there were a few papers, not many. Well, there were hundreds of papers talking about a different gene, and hundreds of others talking about another different gene on chromosome 21, but almost none on this. But you can see there's a bunch of papers where a few people followed up on that original observation by Ruddle, and it was sort of lost. So this guy actually started a company to try to market a protein, an antibody, against, not quite an antibody, but a protein that would suppress interferon action. And the VC people said, no, you can't cure Down syndrome. And so he had to close the company. And so this is all in my presentation, so you can find it. So this was done by Joaquin Espinoza and his group. Kelly Sullivan and Awan Pandey were key members of this team that did this. And this is his thank you slide, which I've added his face to. So thank you for listening to this. I love talking about this research. It's fun science to think about it and do, and it has the potential to make a lot of people's lives better. So finally, I'd like to acknowledge Larry, Tom Check, Leslie Leinwan, Michelle Witten, John C., almost all of whom are sitting here in the audience today, for conspiring to put me into this position. I know what happened. And Joaquin Espinoza, who is currently in Argentina, for agreeing to take on the position of Associate Director for Science and then doing such a great job. And thanks for listening.
So let's take a couple of questions, and then we can take a break. I see a hand right there.
That was a good talk. I work in Somologic, and we do this unbiased proteomics biomarker discovery with thousands of proteins, right? 3,500, you have done this study. But I was thinking, have you thought about, because the genes those are present on chromosome 21, if you do just the soma scan only on those 300-something genes or the proteins, would you clear up and get better idea about the proteins? Those are the clear-cut targets for.
Well, I don't know. We haven't done that. There's only 50 of them of the 300. Only 50 are on the array, and of those 50, 10 of them are increased. None of them are decreased, so I don't know the answer to the question, but it's not a high priority experiment for us because we think the important things are the proteins that are turned up or down that are the result of the signaling that's gone awry. Can we get this question? Charles?
So I'm really struck by the increased heterogeneity in gene expression in the
Yeah, so are we.
T21s. And so my question is, so T21s are intrinsically genetically more heterogeneous because they have two different types of heterozygotes. And you think, is that what's causing this?
I don't know the answer to the question. It might be, but my guess would be no, that what's causing it is simply the higher level of variability caused by induction of the oxidative stress response. So that is a very variable response, and the response to it is also very variable. So that'd be my guess.
So one here, and then Jean, and then we'll take a break.
Thank you, Dr. Blumenthal. I used interferon when I was treating my hepatitis back in the day, and one of the things I learned over the years was that you don't want to be using any type of marijuana, or that now it's legal, but back in the day it wasn't, because it lowered your interferon rate. And I was just wondering, they're using some of the CBDs and CBNs to help children with epilepsy with their seizure disorder, and I was just wondering if there might be something in the THC or in the CBDs, the cannabinoids, that might help with children that are in utero or after they're born, that might have the extra chromosome 21 syndrome or the Down syndrome. If that's kind of clicking with you, in other words, would the THC, the cannabinoids, reduce the interferon present? It's like a lot of people nowadays, if you have a cold, you don't want to be using medical marijuana because it's reducing your interferon in your body, and that's helping you fight off a virus or a pathogen. So if that's clicking at all, thank you.
I'm not sure I get the question because I couldn't hear all of the words you said. Were you talking about epilepsy?
Well, basically, would THC help to reduce the amount of interferon in
THC? Oh, I have no idea. Having never tried it myself.
That was a wonderful talk. I wonder whether the prevalence of prenatal testing for Down syndrome allows one to have some very interesting material to look for the earliest manifestation of the cascade you're hypothesizing?
Theoretically, it should, but as far as I know, nobody's exploiting that opportunity. But, I mean, people are certainly exploiting the opportunity for prenatal testing, which is now has changed the landscape of Down syndrome quite a bit, as you might expect. But nobody's gone and said, let's look at these fetuses that get aborted as a consequence of this. As far as I know, nobody's doing that. But you know, the idea of doing this prenatal testing for some people is probably termination of the pregnancy, but for a lot of people it isn't. It's knowing in advance that you have a child with Down syndrome and therefore getting some knowledge about it. So that's a double-edged sword.
Thank you very much. So let's take a short bio break and